very universal, so you can basically use it on any other proteins as well. Now, with the secondary antibodies, which are already linked to oligonucleotides. Now, these secondary antibodies, if they are close together, you basically have then circular DNA amplification. And upon the addition of fluorescent probes, then to the complementary oligonucleotide sequence, you get fluorescent dots. And each of these fluorescent dots is representative of two proteins in close proximity to each other in a biological setting of 40 nanometers. So this is the technique that we used to look at our limb sections. So we basically did some limb sections, and we started to look at PLA in itself. So the first panel basically shows the test sample where you have two tagged alleles, both MSX and GLI3R present. And in these sections, you have either the absence of the GLI3R allele or the MSX1 HA allele. So in the presence of both these alleles, you have very nice fluorescent dots, which are indicative of the fact that both MSX1 and GLI3R are interacting, or at least in close proximity in a biological setting. And this is a very nice in vivo experiment as well. Now, in the absence of these alleles, you had no fluorescent dots, which were indicative of the fact that the PLA was indeed showing a true protein level interaction in vivo. Further, I would like to stress again that the MSX1 expression is very restricted to the boundary of the developing limb as well. So when we did cross-section and transfer sections of these limb buds and we started to look, we saw that the fluorescent dots were also very restricted to the place where MSX1 expression is expected. So this in itself served as an internal positive control to see that indeed these fluorescent dots were representative of potential protein level interactions. And also, the PLA reaction in itself is concentrated at the periphery of the nuclei, which was also something that has been published before, so it was in accordance to published data as well. Now, having these data then allowed us to look to see how do protein level interactions happen in vitro. So we started to transfect HEK293 cells with cDNAs tagged for GLI3R flag and MSX1HA. And we're able to show that in the co-immunoprecipitation with the anti-flag, we're able to show that the antibody pulls down indeed MSX1. So GLI3R was able to pull down MSX1, and the reverse was also possible where we used an anti-MYC antibody for the co-immunoprecipitate, and we're able to pull down MSX as well, GLI3R as well against the MSX1 antibody. So this basically demonstrated that it is possible to cross-precipitate both MSX1 and GLI3R proteins indeed. So with these experiments, we then knew that MSX and GLI3 are interacting at a protein level, in vivo as is demonstrated by the PLA, and also in vitro as is demonstrated by the co-IP and Western blot results. So we then knew that there is genetic level interactions and there's a synergy which is independent of Sonic-Kitchell, and also this is happening at a protein level interaction. So then, are they regulating then similar functions during limb development, and do they share common targets as well? Now, one of these functions that I mentioned, which is very specific to the anterior part of the developing limb, is the anterior apoptotic domain. So when we look at the controls, as you see here, there is this anterior apoptotic domain, which is completely abolished in the MSX1 to null mutant context. So you have complete loss of this anterior apoptotic domain. And this is exactly also what happens when you look at this GLI3 null mutants as well. So you also lose this anterior apoptosis as well. So indeed, when MSX and GLI3 are mutated, you lose this anterior apoptosis. Thus, both these genes are required in some part for anterior apoptotic processes. And this might then also, in by part, explain this overgrowth and polydactylous phenotype that we see in these mutants. So there's also this similar effect and similar function they're playing a role together in. So then there are further questions. Are they sharing then common targets in implicating these roles? And one of these techniques that we wanted to use to look at this was to do a transcriptome using the MSX mutants. But this then posed another question. What type of mutant should be used? We used the conditional MSX1 mutant for the transcriptome, mainly the MSX1 CRE-ERT2, CRE-ERT2, and the MSX2 FLOX mutant. So to define exactly when to inject this, I did a series of injections from E6.5 to E11.5, and it was really at E8.5, injections to E8.5, which provoked a mutation that showed very high modulations of changes due to the inactivation of MSX in itself. So it was really at tamoxifen injections 8.5 that we started to see striking anterior defects because of the inactivation of MSX. And we used this as the rationale to then inject all these mutants at E8.5 and to collect the RNA at 11.5. Now, also injecting them later stages at E10.5 evoked slight anterior morphogenetic changes as well, which indicated that MSX is important even at later stages in limb development. So with this, we then formed the rationale to inject at 8.5. We collected the RNA of whole forelimbs and whole hind limbs, and we sent it to RNA sequencing to Oxford Gene Technology. We did get back all the results, and we had many genes which were upregulated and downregulated, which we know which are involved in limb morphogenesis. So it was very nice to see that many genes of FGF factors, HOX, HAN2 were upregulated, and many genes such as ALX4, GATA4, and BMP were downregulated. Now these are genes involved in limb modulation, and we were very excited to see that. But what was most interesting for us was the genes involved in proliferation. And these were highly upregulated, such as CDK6, JAK1, and MYCN. But these were the targets of MSX. So these were targets that were upregulated in the absence of MSX. But are they common targets of BLE3 as well? So to do so, we did a comparative analysis. So now we had the data of the RNA-seq, which showed the targets that were up or down regulated in the absence of MSX. We then compared it with Steve Vogel's data, where he mentions the direct putative binding targets of BLE3. So this was an excellent source of information where we could just compare the targets and see the common targets between the two data sets. We came up with 4 main targets which were of interest: CDK6, JAK1, SAL4, and SAL1, all of which are also known to be involved in proliferation as well. Now, so SAL4, as you see, is not expressed anteriorly, and it's known as a common target between MSX and BLE, as I mentioned, and it's not expressed anteriorly in the